morning guys. Today we are going to complete the meiosis 1 part. In the meiosis 1, some phases we had discussed that are prophase 1. Prophase 1 consists of leptotin. One more time, I am recapping here. Zygotin, packaging, diplotin and diakinesis. So in the last class we gone with diakinesis. In the diakinesis, nothing but disappearance of the nucleic membrane, nucleolus and centrioles are moved towards the poles, and next phase it translates to the metaphase 1. So that after the diakinesis, what will be the director? Metaphase 1. We have to mention metaphase 1, otherwise it will be considered as mitotic division. Mitotic division has only one metaphase, but meiotic division has two metaphases. That's why metaphase 1 and metaphase 2 will be known. Next, after metaphase, anaphase 1. Just like that metaphase 1, anaphase is also step 1. In the meiosis 2, anaphase 2 will be there. And lastly, telophase 1 will be there. So today class is about these three. The result, two haploid cells. But this is not the final. These are two haploid cells will go to next stage of the meiosis, that is meiosis 2. So find what is happening in all the three, that is metaphase 1, anaphase 1 and telophase 1. Let us have a look. In the metaphase 1, the bivalents are arranged at the equator. So let us have the nucleus, not that was a cell, it is the nucleus. Here, before this, in the prophase 1, chromosomes are condensed, of course, and also crossing over taken place in the packaging because of the recombination. Later, genetic materials are exchanged. Such homologous chromosomes, along with the chiasmata, they are scatterly arranged in the diplotin and diakinesis. Later, that is the completion of the prophase 1, continued by metaphase 1. The very first in the metaphase 1, bivalents. What are the bivalents? This is the bivalent and this is a bivalent. So this is tetrad. Fine. So these bivalents or tetrads comes to the equator. Before that, they are scatterly arranged. Scatterly means what? Unevenly, not an uniform. So now they had come like this. How many sets of chromosomes will be there in the human? 23 sets of chromosomes are there. All are homologous chromosomes. Clear? This is how, if we take for assumption 4 chromosomes, then all the 4 comes at the center. And that process is called congression. What is happening in the congression? It is a phenomenon in which all bivalent sets of chromosomes, maybe 23 or 50 or 100 or 2 or 4 or 5, whatever. But all sets of chromosomes along with their bivalent shapes comes at the center and form metaphase plate. Metaphase plate is the result of congression. Understand? Now, continued by the microtubules are forming from the poles. Which poles? Opposite poles. So this is one pole and this is one pole. From these, spindle fibers are coming like this. This is what? Next initiative in the metaphase 1. In the metaphase 1, very first initiative is bivalents are coming to the equator called congression. Congression is followed by the formation of spindle fibers. Where does these spindle fibers are coming from? From the center or sides or poles, upper and lower sides. They are opposite poles. So from the opposite poles, microtubules, also called spindle fibers, are coming towards the homologous chromosomes. HLC means what? Homologous chromosomes. So these are homologous chromosomes. This direction I am telling you like this. Clear? From this pole towards homologous chromosome, as well as opposite pole also providing the spindle fibers towards the homologous chromosomes. That is second initiative. After bivalent formation at the equator called metaphase plate, once metaphase plate is formed, spindle fibers are formed from the opposite poles towards the chromosomes. What next? Spindle fibers are forming, they are having three kinds of alignments. One is chromosomal spindle fibers. Chromosomal spindle fibers are also called kinetochore spindle fibers. Kinetochore spindle fibers means they are starting from the centrioles or opposite poles. They are directly connecting to the kinetochore. What is kinetochore? This is kinetochore. So they are coming directly and attached here. A spindle fiber may be aligned like that. Otherwise, a supporting spindle fibers, they are also called continuous spindle fibers. So they are coming like this. They are coming like this because they are giving some rigidity, mechanical strength. Clear? Suppose from the opposite poles or one side, from this side, 10 spindle fibers has come. But here how many chromosomes are there? 4. Out of 10, 4 will go and attach it to the 4 chromosomes. Other than 4, 6 were there. These 6 are giving the strength. Because of their properties and alignments, entire the metaphase plate having some strength in it. Now they are called supporting or also called continuous. Other than this, if between them any are there, just like spindle fibers which are coming from them but not evenly connected, so they are called intergerminal between these two. So there are three types of spindle fibers are there. Number one, chromosomal. They started from the opposite poles, directly connected to the chromosomes. Which part? Kinetochore part, center part of the chromosome. The second one is the supporting spindle fibers. They are coming from the opposite poles but not connecting to the chromosomes. Centromere, nothing but a kinetochore. They are away from that but providing the mechanical strength. That's why they are called supporting spindle fibers. The third one is intergerminal other than these two. So this is what significance happened in the metaphase 1 after the diakinesis, nothing but the last step of prophase 1. So prophase 1 and metaphase 1 are completed. Next, anaphase. What is anaphase? In the anaphase, the metaphase plate going to be split. It becomes half. Direction will be opposite. Let us see. In the anaphase, homologous chromosomes are going to be separate. That is what it is called anaphase. Ana means what? Movement. So movement of chromosomes from center towards their opposite poles is called anaphase. So that segregation happens. That is why this step is called segregation step. Segregation means separation. What are going to separate? Chromosomes. But here the chromatids are remaining same. Number of chromatids are 2. Suppose if we here one pole and there is an opposite pole. From here the spindle fiber is there and this is the kinetochore. From here there was a 2 chromatids. So the rest of chromatids are same. Like that. And same here. One and a 2. Reduction taken place. So metaphases plate completely split, then they become half. That's why it is also called disjunction. Metaphase plate is going to be split and form disappearance of that junction. So that is called disjunction of chromosomes. Disjunction of chromosomes takes place in the anaphase 1. It is also called phenomenon of segregation of homologous chromosomes. Now, division of centromere is absent. Very, very important. What is centromere? In entire the chromosome, this is centromere. This is centromere. Centromere will not be divided. It will be doubled. Understand? Now, this phase is responsible for the reduction of chromosome number. CN means chromosome number. Chromosome number will be reduced. Means diploid become haploid in the anaphase 1. Very, very important point here. Okay. Then the daughter cells will have haploid nucleus. Till that cell is in diploid. In the anaphase 1 only daughter cells become haploid. Once a complete segregation of chromosomes are happened in the anaphase 1, then we can call up that is karyokinesis. Karyokinesis means what? Karyo means nucleus. Kinesis means division. So division or separation. Separation happened means division happened. So, but nucleic membrane is not formed. That is the drawback in the anaphase. That's why we cannot say in the anaphase karyokinesis doesn't happen. What happened then? Chromosome segregation only done. Chromosomal segregation is followed by karyokinesis. Means here a set of nucleus, here a set of nucleus is formed. Then here segregated chromosomes were there. Here segregated chromosomes were there. So what is this? This is a nucleic membrane. This is also nucleic membrane. And this nucleolus is also formed. Nucleolus is a cell organelle that present inside the nucleus, part of nucleus, which helps in the ribosomal synthesis. Without nucleolus, ribosomes cannot be produced. We can say there is a factory of ribosomes. So it's a characterization of eukaryotic cells. Then if it is the cell, then you can see two nuclei are there. This stage is called dyad. Dyad is the stage in that one cell consists of two nucleus. If you imagine nucleic membrane is not there, nucleolus is also not there. This is the phase which is indicating anaphase 1. After anaphase, what is happening? Reappearance of nucleolus and reappearance of nucleic membrane. So this is the nucleolus and this is the nucleic membrane. Then these are two separated nucleus in one cell. That condition is called dyad, binucleated state. Understand? But the important thing is these both the chromosomes are with new characters, variety of characters because of the recombination crossing over taken place in the prophase 1. Which phase? Cytokinesis. Pack 18. Understand? Now, so karyokinesis is followed by the cytokinesis. Means here there are the two nucleus were there inside that chromosomes and nucleolus is also there. This is karyokinesis followed by cytokinesis. Means two cells. It is two nucleated cell. One cell that with the two nucleus, whereas two different cells with the nucleus. Then this step is called telophase. Telophase means what? Karyokinesis followed by cytokinesis. Cytokinesis means cell division. Karyokinesis means what? Nucleus division. After that, we can say meiosis 1 is completed. Meiosis 1 result two haploid cells. Chromosome number become half, reduced. In which step? Anaphase itself. So, the completion of meiosis division 1 taken place and it leads to meiosis 2. Here one should understand that before any division, cell should be prepared. For the mitotic division M phase, cell is prepared about 24 hours. And entire the cell cycle is 24 hours. 90%, nothing but 23 hours cell prepared and 1 hour cell divided. That is why mitotic division given 2. Even in the meiotic division also cell is well prepared. But here two phases are there. Does the cell was prepared for meiosis 1 or meiosis 2 or both? Actually, cell was prepared for meiosis 1. Result, two haploid cells. After that, it is immediately entered to the meiosis 2, it will be failed. So that cell again has to prepare for equal division. That step is called interkinesis. interkinesis. So inter means what? Gap, between, a space. Kinesis means what? Division, between two divisions. What are the divisions? Meiotic division 1 and meiotic division 2. There was a short-lived space. This is called interkinesis, is a short period for cell. Here it will be prepared very quickly for the equal division. Why I use the term equal division? Meiosis 2. Meiosis 2 receives a cell which is a type of a haploid and which provide two haploid cells. Equal types of cells are coming. That is why it is called equal division. Whereas meiosis 1 is reduction division. Here a diploid cell went and two haploid cells were formed. So 2n become n and n. So here reduction taken place, but here not. So in the next class we will discuss about how the meiosis 2 happened and result how many cells.
Meiosis is a nuclear division that occurs in the germ cells of sexually reproducing organisms during the production of reproductive cells called gametes. Meiosis is a reductional division that reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis 1 and meiosis 2. The entire process of meiosis results in the formation of 4 haploid daughter cells with half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome number gets restored. Meiosis 1 is the reductional division in which the chromosome number gets halved and distributed between two daughter cells. Meiosis 1 includes 4 stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. Let's know about the homologs before studying the phases of prophase 1. Each diploid cell has 2 copies of chromosome, one inherited from the male parent and the other from the female parent. These 2 copies, coming from different parents, are similar to each other and are called homologs. Now let us look at the different stages of prophase 1 in detail. To begin with, let us learn about leptotene. During leptotene, the chromosomes condense and become visible. The zygotene phase is followed by pachytene. During pachytene, the chromosomal crossing over now takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination modules to be visible. As the crossing over completes, it marks the end of the pachytene phase. Diplotene is followed by diakinesis, which is also the last stage of prophase 1. During diakinesis, the chromosomes are fully condensed, the nucleolus and the nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase 1 is followed by metaphase 1. During metaphase 1, the microtubules from the opposite poles attach to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate with a random orientation. Either of the parental homolog takes up any site on the spindle. The metaphase 1 is followed by anaphase 1. During anaphase 1, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules. The sister chromatids, on the contrary, remain attached at the centromere. In this module, you have learned meiosis is a reductional division which reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by 2 cell divisions, meiosis 1 and meiosis 2. Meiosis 1 includes 4 stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is further subdivided into 5 stages: leptotene, zygotene, pachytene, diploidine, and diakinesis. During leptotene, the chromosomes condense and become fissile. During zygotene, the duplicated chromosomes pair with homologs to form bivalents. During pachytene, the attachment of homologs to each other along their length, that is synapsis, is complete. In diploidine, synapsis brings to breakdown, the chromatids start to pull apart, and chiasmata are visible. By diakinesis, the chromosomes are fully condensed, the nucleolus and nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase 1 is followed by metaphase 1, when the microtubules from opposite poles attach to the chromosomes at the kinetochores and the bivalents align along the equatorial plate. During anaphase 1, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules, forming 2 haploid sets. During telophase 1, the chromosomes reach the poles and nuclear envelopes are reformed around the 2 daughter nuclei and the nucleoli reappear. A furrow appears in the cytoplasm and the cell divides into 2 by cytokinesis.
But in this session, we can say meiosis 1 is completed. What is the phase that is indicating between meiosis 1 and meiosis 2? Cell must go on through the interkinesis. Hope you understand the concept. Enjoy the assignment.